novelty detection is fundamentally important, not just to human beings, but to essentially every animal with a nervous system. And so to detect novelty, you have to have a way of encoding familiarity. It's just that simple.
Is there a path to, for example, taking a drug that would ensure that whatever you're doing at that moment, you'll remember?
Yes.
There is.
Yes.
Because that would be extremely convenient for a lot of things, you know?
It is.
Forgets.
I mean, that would be or forget. Yeah, exactly. If you could choose.
Yeah. Forgetting is probably more therapeutic. But yes, the answer is yes. In fact, there was a big push in the 1990s, late 90s, early noughts, to develop memory-enhancing pharmaceuticals. It was called the memory space in biopharma. It's like, we got to get into the memory space, right? And develop drugs. And the whole concept was to take a drug that would actually boost the actions of these biochemical pathways that can convert a temporary change into a permanent change. And there are drugs they'll do this. But the problem is that every drug that I'm familiar with has side effects that make it not therapeutically useful. They can still be abused.
What's like an example of the drug and the side effect, and like how does it work?
I don't want to give examples that sound too benign and promote the use of these drugs. You have to look that up. But one of the key steps in converting a temporary memory into a long-lasting memory is the generation of a second messenger in neurons called cyclic AMP. This cyclic AMP turns on a pattern of gene expression that is known to regulate the persistence of synaptic plasticity. So one of the early attempts to develop memory-enhancing pharmaceuticals was to develop drugs that inhibit the enzyme that degrades cyclic AMP. That's an enzyme called phosphodiesterase. And in animal studies, it can be shown that, yes, manipulating phosphodiesterase can really profoundly modify persistence of memory. The problem is that the drugs used in humans caused terrible gastrointestinal effects. So as we joked, it seared the memory of you sitting on the toilet because it was not a great experience. And they couldn't work their way around the side effects to be viable.
Is that related to like the delivery? So if you take it orally, maybe versus taking it, I don't know, intraspinal?
No, it wasn't. Well, yeah, maybe if you took it in direct injection into the brain, you might get away with it. But actually, there may be I don't want to call it a happy ending to the story, but there's a specific isoform of this enzyme that was the sort of thought to be maybe the magic bullet because it's expressed highly in the brain and not in the GI system and so on. And it took a long time to develop small molecules that could selectively target that. And I think there has been a breakthrough in that. In fact, they are in clinical trials now.
And so that would be like for dementia probably, right?
I think that the big objective is develop a drug to help people with dementia, age-related memory impairment, Alzheimer's disease. The studies that they're doing, and maybe we're going to talk about this at some point, actually more advanced studies are done on a rare condition called Fragile X syndrome, which is a form of intellectual disability. There's really a different rationale for using these drugs in Fragile X than the one I just gave you, but I think that it's being used as a test case to assess safety and things like that of the drugs. So we'll see. We'll see what happens.
Does this somehow correlate also to people who have, these almost like pathological photographic memory, like the kind of people that can look at one picture of Manhattan and then draw it from memory?
Yeah, amazing, isn't it? I mean, truly amazing.
Yeah.
And that is a debilitating gain of function. Right, is that you can't get rid of some of this.
But are they biochemically, for example, like is that enzyme broken or like something like, has it actually been figured out, like how they're, what's
I don't know the answer. I don't know the answer. And I'm not even sure the question has been asked because it really depends on what path you've taken to get to the question. I would say that'd be extremely interesting to understand that, whether there's a clear biomarker of that type of persistent and unusual plasticity.
So the plasticity, as you mentioned, is an ability of brain to modify itself after certain experiences. And in terms of our visual cortex, the main experience we've been talking about is to deprive the vision. What other major experiences we can have in life to modify our visual cortex?
Great question. So we've been talking about monocular deprivation because Hubel and Wiesel got this game started in 1963. And these experiments are still happening all around the world as a paradigm to study experience-dependent plasticity. And one of the reasons, there were a couple of reasons why this has been pursued so intensively by generations of scientists now. And one is that, well, it's a question of brain development. It's a question of how synapses form and are modified by activity. So it's a very fundamental aspect of brain development and whether we could gain insights into that. A second reason that it attracts a lot of interest is it actually models a human clinical condition that we may talk about later, but called amblyopia, which is a condition where children are born with unbalanced vision in the two eyes, and the brain doesn't wire up correctly, and there's a permanent visual disability unless it's treated early in life. But the third reason that it's attracted a lot of interest is that, at least at the time, it was the first robust demonstration in a mammalian brain of experience-modifying connections in a way that was relevant to the synaptic basis of learning and memory, or believed to be relevant to the synaptic basis of learning and memory. These studies attracted the interest of people involved in neural network models, computational models, trying to understand how networks of neurons could modify their properties based on input through synaptic modification. And I thought, I'm an enormous fan of the Bienenstock, Cooper, and Monroe theory of synaptic plasticity. And, a tribute to Leon Cooper, who died recently, who was the esteemed theoretical physicist for his work on superconductivity. But in neuroscience, he's also well recognized for developing this theory of synaptic plasticity. And what motivated him really was to understand the synaptic basis of learning and memory. But he tied his theory, or tried to anchor his theory, to experimental observations in visual cortex and the effects of manipulating visual experience early in life. And I should just say he was a mentor of mine. I trained with him, and I had a huge impact on my work. But to sort of get to the question that you originally asked, which is what other types of experience modify visual cortex and so on. Of course, our visual systems are modified by experience. That's how we're going to remember. We'll be able to recognize one another when we see each other in the hallway next week, right? We're forming visual recognition memories. That's happening in the visual system. But for years, it was thought that the primary visual cortex, the part that we've been discussing so far, is more machine-like in its function. And so that, you do some early programming work based on visual experience to get it tuned up, but then it's frozen in place, and then that information is distributed to the other 20 areas of cortex that are devoted to analysis of the visual world, right? And that's been a prevailing view. I think that in recent years, it's become more and more apparent that there's still a lot of experience-dependent plasticity in the primary visual cortex. And I think that we probably need to be specific there to say, what species are we talking about when we say that, right? Now, there are examples in the human visual system of experience-dependent plasticity that seems most likely to occur in primary visual cortex that serves things like perceptual learning. Why with training, for example, you can discriminate visual stimuli that otherwise you couldn't. So, with practice makes perfect, and there's some fine-tuning of connections in the primary visual cortex that serves that. But as you know...
What's like the paradigmatic example of that?
It would be, for example, I'm trying to think, there was a researcher whose name I do remember, named Avi Carney, who did some really important work on this. I think they were orientation discriminations, the ability to discriminate very fine distinctions in the orientation of line segments. And that could be learned. And one of the interesting things about that was that the learning would pop out after a night of sleep. So it interfaced with the role of sleep in plasticity. But, that's...
Is there a certain type of sleep that gives you that the learning?
Well, all sleep is good, but it appears that, it's such a wonderful question. And such an exciting area of neuroscience, honestly. We think about really two phases of sleep, two broad phases of sleep, slow wave sleep and REM sleep, rapid eye movement sleep. And our brains cycle through these different types of sleep in a good night of sleep. And each phase of sleep is thought to contribute something different to consolidating the synaptic modifications that occur during wakefulness. I'd say slow wave sleep, REM sleep is sort of sexy because it's considered to be dream sleep. We know we have wild experiences. More often than not, they're likely to be occurring during REM sleep. And REM sleep seems to play an important role maybe in like what's going on in adolescence, which is whittling connections down in REM sleep. Slow wave sleep has been a revolution in understanding what's going on in slow wave sleep in recent years. And one of the things that it seems to do is it cleanses the brain, which is important for all synaptic plasticity. But it also, there's a replay of experiences during wakefulness that occurs during parts of slow wave sleep that can consolidate the synaptic modifications. Yeah. But I wanted to get back to this question about experience-dependent plasticity in the context of learning. And I think in a way we have been fortunate, perhaps, that the world has been consumed with studying mouse vision. I laugh because mice have a terrible visual system. I mean, let's face it. They're nocturnal. They have laterally placed eyes. They have very little binocular vision. They have a rather poorly differentiated visual system. It's nothing like the beauty of a primate visual system. Nothing like it.
So who's the next in the chain after mice? Is it cats? Or?
Cats have been used historically, and they have a lot of features that are in common with the primate visual system. But the caveat there is that many of these features appear to be a consequence of parallel evolution, convergent evolution, so that it's not clear that, I mean, cats and primates diverge very early in mammalian evolution. So it may be that we may have all come from a rodent-like ancestor, and cats developed the same solution that primates did, but we have to be a little bit careful of saying, oh, a cat's a small monkey, something like that. It's not that way. But there are other great, I mean, every species brings something interesting to the table. You have to take into account the niche that species occupies. So, let's face it, mice are nocturnal. They have laterally placed eyes. They don't have a lot of binocular vision. They don't really rely on vision. They rely on somatic sensation and the sense of touch, their whiskers and so on. But nevertheless, that's the species we've got. They're intensively studied because we can do genetic manipulations in mice. That's the main appeal of mice. They breed rapidly, have big litters, and we can do a lot of genetic manipulations in mice that have revealed a lot of the mechanisms of the plasticity that we're discussing today. But one of the features of the mouse visual system is that, unlike a human, which may have, I don't know what the count is, but maybe 20 distinct cortical areas that are devoted to the analysis of the visual world, mice have relatively few. So a lot of the functions that you might have to go searching high and low for in a human are also being accomplished in the primary visual cortex of a mouse. So there are types of learning relevant plasticity in the mouse visual cortex that are rather readily observed and easily studied. So the mouse has been great that way. I'll give you an example from our own work. One of our objectives here is to monitor the strength of responses from the two eyes, right, in visual cortex. That's the name of the game for us. There are a lot of ways you can do that. There's some very fancy, technically sophisticated ways, but there's some pretty primitive but very powerful ways as well. And one is a phenomenon called a visual evoked potential. So it's not recording the nerve impulses from individual neurons, but it's recording some summed output of a population of neurons. And the way to elicit a visual evoked potential is to show a stimulus that changes abruptly. And with every one of those abrupt changes, you average the signal that you're recording. And if you do that enough times, the average reveals a visual evoked potential. So it's riding on a very noisy background, but you just keep repeating this synchronous activation and you get a nice response. And that's kind of what we do in a lot of visual studies, is you do multiple trials and you average across trials, whatever you're measuring. The way we've been doing it in my lab and many labs is that we have the stimulus it's like a pattern of stripes. We call it a grading stimulus. So we have black and white stripes. And to elicit a response, we do a phase reversal so the white becomes black and black becomes white. So we phase reverse once, phase reverse twice, and keep doing that. You can phase reverse 100 times. And then you average the responses that are time locked to that phase reversal. And that's what gives you a visual evoked potential. So this is the way we monitor responses from the two eyes, right? You with me? Okay, cool. And we can do this through chronically implanted electrodes. So we can put an electrode in the brain of the mouse, and we can plug them in and stimulate him every day. He's awake, looking at this computer screen. And a chance observation that a brilliant student of mine made, named Nate Sawtelle, who's now a esteemed professor at Columbia, he observed that over days, the response to that stimulus got bigger and bigger and bigger. At first, it could get twice as big.
I mean, bigger is this what do you measure? The potential?
Yeah.
So the amplitude gets...
The voltage deflection.
Voltage deflection, okay.
So it could go from 50 microvolts to 100 microvolts. So a huge change in the land of biology. And at first, we suspected it was a technical problem. What are we doing wrong? And we were building the plane as we were flying it anyway, because no one had ever done this type of recording in awake animals before. Nate was doing it. And Nate had the brilliant insight to say, well, I wonder if this is plasticity. And if it is, what would happen if I shifted the orientation of those stripes ever so slightly? So he plugged an animal in. After the animal had seen, say, vertical stripes for 4 or 5 days, the response had gotten big. And he shifted the stripes 45 degrees, and the response was right back down at the baseline. He described this phenomenon that we now call stimulus selective response plasticity. That as one who's devoted his life to studying plasticity in the brain, I've never seen a more robust demonstration of plasticity than this one. No finesse is required. Any student can come in and we can get that experiment to
Did you try to do it for various degrees
Yes.
and see how it...
Yes. And amazingly, as few as 5 degrees off of the experienced orientation, you'll see a significantly reduced response. So it's very tightly tuned to the stimulus orientation. So this is a gift to people that study plasticity. It's like, oh my God, we have this super robust change in the response to an experienced input. And of course, my instinct is to say, okay, let's try to understand the mechanism and do a lot of mechanistic experiments. And so we do that. We unpack it as much as we can. But of course, the deeper question, and the question that a reviewer invariably asks is, so what? What does this have to do with learning? This is a plasticity, yes, but what does it have to do with learning? Then I have to introduce another brilliant student of mine, postdoc, named Sam Cook, who's now on the faculty at King's College in London. And Sam had noticed that when the animals see the stimulus for the first time, they're awake, they would show this alerting response and they would show interest in the stimulus. But as the animal was plugged in on a daily basis to look at the same stimulus, they showed less and less of a behavioral response. And so they would habituate. It was a behavioral habituation. So first, and we could measure this. We could measure their forepaw movement, for example. So we saw this vigorous forepaw movement in the first trial. The days later, no movement at all. Then we shift the orientation and they start moving again. So the animals are behaviorally reporting that they recognize the difference in the orientation. And then Sam teamed up with, I have a whole bunch. I can't give credit to everybody because I have so many great people that have worked on this problem. But he teamed up with another scientist named Rob Komorowski, and they did this in an open field. They showed that if they expose animals now in an arena with a computer monitor showing the stimulus, that initially the animals will run up to the computer monitor, they'll check it out. They're very cute, actually. They're really interested in this stimulus. But then next day, they're like, same old stimulus, nothing good happened, no interest whatsoever. And then on a test day, we can put the novel stimulus on and they show the interest again, right? So then we can plug them in and ask them, okay, well, are you responding differently to these experiences you had in this open arena? And yeah, so there's a big difference in the evoked response. So what we have is a behavioral report of learning, of actually a fundamentally important type of learning called visual recognition memory. And it's also learning to distinguish a familiar stimulus from a novel stimulus. And, a basic decision we have to make every day is whether a stimulus is novel and worth devoting attention to. It's novel because it could be a threat, or it could be an opportunity, or it could be nothing, right? Those are the three options. And so novelty detection is fundamentally important, not just to human beings, but to essentially every animal with a nervous system. And so to detect novelty, you have to have a way of encoding familiarity. It's just that simple. And then in the grand theme, it's visual recognition, how I recognize your face. So we now have this really great system. We have an electrophysiological readout of a behaviorally significant type of learning. And then you do the next level of experiments and you say, okay, do the mechanisms that serve this electrophysiological plasticity also serve the behavioral readout? Answer is yes, they do. So we worked out I had another great student I should mention is Misha Frenkel, who worked also with Nate in the early days to work out the mechanisms of this phenomenon. And then we were able to show that those mechanisms are essential in primary visual cortex for this form of memory. So we're getting closer. We feel like we're getting closer to us, the holy grail of being able to point to the synapses that are actually encoding that type of memory. We're not quite there yet, but we get closer every day.
I'm just more curious from the optics point of view. Did you try to change the spatial frequencies and the brightness?
Yes.
So
yes.
the visibility. I'm more like inspired. Recently, Michael's baby was at our place, and somehow there was like so many cool things around, but it would try. Yeah, the cats were like running around, but then his kid would try to go to the display to
change the temperature on the thermostat.
And I don't know why for a kid it would be display is more interesting than something else happening in the room.
Right.
So I thought maybe it's the brightness, like the acuity, the maybe spatial frequencies. Which affects his brain.
It's attention grabbing, right? What grabs your attention, but the things that attract attention are novelty, but of course, and motion, any moving stimulus attracts attention, and a stationary stimulus. Yeah, it depends. It's so great. Actually, it's so cool, Misha, because it encodes orientation, it encodes spatial frequency, it encodes contrast. So all these features of the visual stimulation are being encoded as a familiar stimulus, and if I change any one of those features, it's seen as a novel stimulus.
Is there one thing, people seem to find like fractals more pleasing or interesting, or sometimes maybe in some ways less surprising because there's so many fractals in nature? That's one reason maybe why the thermostat is like more interesting than the cat, because the thermostat is not a fractal. Whereas the cat actually is mostly a fractal, like it's like a lot of fur, right? I wonder if there's like a fractal inside the brain that like compresses the information. If like you're looking at a tree, it's mostly the same thing over and over.
Exactly. Yeah. No, I don't know the answer, but it's a great thought. It's a great thought.
Did you try showing the mice fractals ever?
No, but I'm going to right after we finish. One thing we did do, one thing we have done is not necessarily related to fractals, but we've done sequences. So I have another former postdoc, Jeff Gavornik, who trained or exposed mice to sequences of stimuli, just different orientations of these bars, but always in a stereotype sequence. So we go A, B, C, D, A, B, C, D, A, B, C, D over time. Yeah. So the animal will just look at A, B, C, D, and then come in the next day, look at A, B, C, D. And then on the last day, he would scramble the order and see what's going on. And so in some ways you go, okay, well, yeah, of course it's going to show a phenomenon not like the stimulus selective response plasticity, what we call in the lab SERP, SRP. But in fact, it turns out to be different from SERP, not necessarily more interesting, but different. And the key experiment, the one that really blew me away, was that after the animal's been trained on A, B, C, D, and remember, we're recording the electrophysiological response, A, B, C, D, right? He shows stimulus A, and he omits stimulus B, and we still see a signal in the brain that is the signature of the occurrence of stimulus B. In other words, I don't know what the animal perceives, but the brain is behaving as if stimulus B is there because it was anticipated by stimulus A. And if he takes another orientation that the animal hadn't learned in sequence, you don't get that, what we call phantom response. So the animal, this is in primary visual cortex. So there's a deeper level of plasticity that's keeping track of time, and the timing of those stimuli mattered. Right. So if he altered the time, he also didn't see the effect. It's a playground, honestly, to do clever experiments in primary visual cortex. And these are all, the experiments that I've described so far are just unrewarded stimuli. We haven't gotten into fancier things like adding reward, which will engage yet other circuits. But this is just passive stimulation.